Everybody, please take your seats. I find myself, like in the beginning, being asked to look after the final session. Charlie Messina, who gave his talk, is now committed to go and run some other meeting somewhere else. So now here I am. So bad news is, it sort of feels like the first session. And Fred gave the opening talk. He'll give the closing talk as well. So we'll be back at the start before you know it. But I think the really exciting part is we kept all the best talks to the last session, didn't we? So it's really a pleasure for me to introduce Kirsten Tusscher. Sorry if I said your name badly. And for those of you who are less familiar with CropXR, I think this is a really exciting talk because we're going to find out a lot more about CropXR. So, Kirsten, I'll hand it over to you to begin the session.
Okay, good afternoon. Hopefully everybody is still more or less awake. As he said, I'm Kirsten Tusscher. I'm a professor in theoretical biology at Utrecht University, and I was asked to present CropXR, this recently started Dutch initiative, to you. And what I specifically want to talk about a little bit is how we envision that we do data integration with mechanistic modeling, machine learning, and control theory to arrive at smart breeding approaches. So I guess CropXR, like many initiatives around the world, have as an overall goal to develop climate resilient crops. And nowadays, most of us realize that crops are all the time exposed to multiple stresses at the same time. So we really try to address how can you develop new strategies to breed for complex traits that give a plant multi-stress resistance, realizing that this will be very polygenic. So just briefly, because this is a rather new initiative in the Netherlands, to introduce this to you. So CropXR is basically a sort of virtual institute, so it doesn't really have one specific location. And it constitutes of 5 different universities in the Netherlands, 5 universities of applied sciences, some biotech companies, and most major breeding companies in the Netherlands. And it has received funding from the Dutch Research Council, as well as from FAR. And well, it consists of a number of parts. So first of all, we have this PlantXR program that I mostly will be talking about, which is the scientific part. And it consists of a sort of fundamental science core, as well as a satellite program where we will be going more towards crop plants and real breeding situations. We also have the AgroXR part, where we will take this even one step further and go into field trials. We have a DataXR part, so we also put a lot of emphasis on where do we store the data, how do we make the best use of the data, how do we make sure that all the data is well annotated, etc. And then we have this EduXR part, so one of the efforts that they are driving within this part is to set up a new minor here at Wageningen University, but accessible for students across the Netherlands, but that have different backgrounds, and then get this sort of training in this interdisciplinary approach. And then there's a final part, TransferXR, to hope at the end of this 10-year period to already be at the stage where we can transfer things to actual applications. So PlantXR is really the research-focused part of CropXR, and sort of our mission statement is there to develop what we call a smart data approach for breeding multi-stress resilient crops, and to do this by having a very interdisciplinary approach. Now, smart data is, of course, a strange thing to say, because data in itself cannot be smart. We can perhaps be smart in the way we use it. That's already one thing. But within PlantXR, we want to take this one step further. We want to also be smart in deciding which data are we going to generate. We're living in a time of big data, but it's very important to realize that big data are not for free. They're not for free to obtain, either in terms of time and energy and money that costs. It's not for free in terms of storage and how much energy that costs us. I think all our green goals go out of the window just because we have all these Google centers that take up our windmill energy. And they're also not for free to train the models on or to put queries on. And now all of our children in high school are using ChatGPT to write their thesis instead of Googling it. And I think, they can all become vegetarian, but maybe it's more easy if they just keep using Google. So what about this demonstrator work package? So here it becomes maybe a little bit uncomfortable because I'm at a sort of crop breeding symposium, but we're actually going to start with Arabidopsis. So why? So what we did here is we said, okay, this is the work package that all the other work packages have to learn from and that they are sort of waiting for. So we need to have something where we can hit the ground running. So let's go for two very climate resilient stresses, heat and drought, but let's begin humble. Let's go for Arabidopsis simply for pragmatic reasons. It has a rapid life cycle, we have great genetic tools, we can have relatively simple phenotyping because, well, sometimes you can just grow them on an agar plate and visualize their root system, and we have a lot of prior knowledge. And so we're going to set up this sort of work chain where we have existing data, we are generating a lot of data, and we want to do this hybrid mechanistic machine learning modeling, and then a feedback loop to see like, okay, what new data do we want? So what about this smart data approach? Well, our idea is that if you go, the resolution seems to be really awful. So our idea is that if we go for this hybrid mechanistic machine learning modeling, you can really look at the genetic network, so at the molecular underpinnings, and see how is the decision making taking place in the plant in response to these stresses. So we can also clearly see where are trade-offs happening, where is information integrated, which knobs might there be to turn. And we hope that by having these mechanistic insights, you need fewer data than when you would just use a black box approach. Another advantage of these mechanistic models is that you can use formal control theory, so you can identify where do these models have their weaknesses and uncertainties, and use that to identify what would be the most informative next experiments to do. So rather from big data just because we can, smart data because that might be wise. Now, I'm very closely working together with the translator work package, and there the idea is that the methodologies that we've developed for Arabidopsis, we're going to translate to a subset of relevant crop species. And here again, this smart data approach comes in that our idea is let's first take the Arabidopsis model, let's then do comparative genomics, so that, for example, we can see that another species has more or less gene copies of a certain important gene, or that a binding site or an interaction is missing or is extra, so that we can sort of make a naive crop model from the modifications of the Arabidopsis model, run it, compare it to that species, and then see where differences are, and again, inform what is the most urgently needed data, because clearly it's much less easy to get all this data from the crop species than it is for getting it for Arabidopsis. So, well, to link this up a little bit to what you guys been discussing here the last 2 days. So what are we going to do in terms of genotype and environment times phenotype mapping? So on the one hand, we will do in-pot experiments on Arabidopsis, so exposing them to a matrix of 3 different temperatures and 3 different drought regimes, where we will look at the Columbia 0 ecotype that is most used as a wild type, but also look at key mutants that we know already play an important role in how well adapted Arabidopsis can be to temperature and drought. And of course, over the longer run, we want to identify new genes that are maybe specific for this combination of stresses. And at the same time, what we are doing is that we're taking a set of accessions of Arabidopsis that have been gathered all around the world, and that have been evolving for different environmental conditions. And so we cluster these environmental conditions, and we can really see where they correlate also with different temperature and drought regimes. And for each of these different sort of combinations of climate and weather variables, we took subsets of 15 to 20 ecotypes, so that you can also see maybe species evolved under similar conditions, but have evolved different types of adaptations. And so we're going to do phenotyping on these for the high throughput experiments on the plates. Those are being done in the NPEC, so the National Dutch Plant Phenotyping Facilities that we have in Wageningen and in Utrecht. And we're, for example, developing machine learning approaches to do root system architecture phenotyping. You would think that would be easy, but Unfortunately, small roots that are almost transparent also have disadvantages. Of course, we do also all kinds of phenotyping, like leaf surface area, fluorescence, to see how photosynthesis is doing, and measuring things like hypocotyl length, root length, etc. And then, of course, for the different mutants, we can already clearly pinpoint it. Well, this gene was different. And for the ecotypes, we will link the phenotypes to the GWAS analysis. But then getting to mechanism, so because that's really what we want to aim for in this project. So we want to arrive at sort of the molecular control networks that drive plants to do morphological and physiological adaptations to stresses. And for that, we're going to do a lot of transcriptomics and metabolomics measurements in these experiments. And those will be mostly done on the pot experiments. And as you can see, for the first day in the experiment, we're going to take a very dense sampling, but then for later on, it will be less dense. So what did we decide to look at? Perhaps maybe not the most agronomically exciting properties, but let me try to explain you why we chose Arabidopsis seedlings and why we chose these responses. So we know that in response to temperature, plants, they elongate their stem, their hypocotyl, they do this hyponasty, they put their leaves upwards in order to cool themselves. In response to drought, a very clear response is that they elongate the root system, and then temperature and drought also have impacts on stomatal aperture. So what is nice about these three different things is that root elongation seems to be promoted by both higher temperatures and lack of water. So here we have a clear convergence. So are these effects additive or even more so? For hypocotyl elongation, there's a clear antagonism. So the water limits how much temperature induces this elongation. And for stomatal closure, it's really that the drought effect even seems to override any temperature effect. So it's not so much an averaging out of these two antagonistic things, it's really one thing overriding the other. And if you look in the literature when it comes to what kind of genes are involved in these different processes, very much the same suspects turn up again and again. So we thought this would be interesting to look at, because then you can wonder, okay, I have similar environmental inputs, I have similar players, yet depending on the process and depending on the plant organ, we get different responses. So how are these things integrated in molecular networks? How is the context of the tissue or of the organ important? And how are these different decisions weighted? So just to give you a sneak peek into the kind of approaches that we are developing here. So for now, we started with very simple single plant models that consist just out of 3 compartments. And then if you look into one of these compartments, we really look at the molecular networks of the genes that are playing a role here and that we know of that they are responding to temperature and drought. And while experimentally we are mostly collecting transcriptomics data, some metabolomics data and some other stuff, in the models we are really trying to look also at protein levels, whether proteins get activated or deactivated through phosphorylation, whether they form complexes, whether maybe some molecular condensates are being formed in response to different water or temperature levels, etc. So we really try to go down to the molecular mechanisms. And one of the nice things here is that, well, you can do, of course, a lot of reading because there's also a lot of hypothesis-driven research on individual genes. But ChatGPT and things like that can also be put to use in these kinds of contexts. So we have something like the plant connectome that people have developed where they use GPT-based techniques to browse all publicly available abstracts, and that you can then use to build knowledge networks between genes, between processes, between hormones, between environmental stresses, etc. Of course, you still have to curate that, but this helps us to enhance these models. And important in these models, we're also considering the communication in terms of signals that are happening between the different plant compartments and how these also depend on environmental conditions to see which parts are sort of concerted actions, which parts are organ-specific autonomous actions, etc. So how does this then look like? Well, typically we use differential equation-based modeling. Of course, at some point you can consider adding stochasticity or something. And we just model the dynamics of these different factors and how they influence each other in these networks. And we translate that then also into, for example, how is the hypocotyl growing, how is leaf area growing, how is root length growing, and how does that depend on the conditions that we simulate which affect certain of these players in these networks. And what is hard to see here, the light gray has turned really, really light gray, but supposedly we incorporated here light and dark cycles. And we still also are working on incorporating things like circadian rhythms. And then what do we do with these models? Well, typically we look at these endpoints and we compare it, for example, with these experimental studies showing that indeed in high temperature you have a longer hypocotyl, but if you simulate drought by adding the ABA hormone, this effect is strongly reduced. So here you see the very different bars and here they're almost equal. So then we use these models to see, like, is what we now have in these molecular models, is it sort of necessary and sufficient? Can we simulate mutants? Can we simulate intermediate conditions, etc.? And of course, the models, we only just started. Our PhD is, well, I think, working with us 4 or 5 months. So clearly, we are in the very early stages with this modeling. So, well, these are just very simple compartment-based models with very limited physiology in it. But when I try to talk to people that are mostly into bioinformatics or into machine learning and just want to look at transcriptomics data, I always tell them that this is not a simple input-output system. It's not like I plug in a temperature and I plug in a water level and then I press a button and then all the transcriptional dynamics roll out. This is a feedback loop, because in the end what is relevant for the plant is, of course, what would be the internal water status of a leaf or what would be the internal temperature of the leaf. So that's what your photosynthesis efficiency or something would respond to. So it's important to take into account that if the plant opens or closes its stomata, that has consequences for the leaf temperature, which then has consequences for photosynthesis and the amount of carbon it has available to also, for example, elongate the root. So we want to, in the end, move our molecular networks into functional structural plant models to sort of close this feedback loop and to show that the transcriptomics dynamics that you're measuring are both cause and effect, because they are responding to your temperature, but they're also driving these responses. But as said, we are only in the early stages of this. But I also said, well, we don't just want to do mechanistic modeling, because the nice thing about mechanistic modeling is, of course, that you can understand why a behavior emerges. But this mechanistic understanding usually works well if you keep your model at least within some bounds. So typically when we do omics, well, each and every experiment that you do, you will find at least 5,000 genes changing their expression. So that's typically not the kind of dimensionality you want when you're developing a differential equation-based model, because simply you wouldn't have enough data to fit all these parameters. So what we want to do is we want to sort of marry this with mechanistic modeling with machine learning modeling, which sort of has the opposites strengths and weaknesses, right? It can deal with a large amount of data. It can be very quantitative and predictive, but typically it's black box or gray box, so it offers limited insight in why things are happening. So what we want to do is to see if we somehow can bring these two approaches together. So we're still early on in this process, but right now we're exploring a lot of different strategies. So for example, with people at Delft University, we're looking at dynamic mode decomposition in Gaussian processes. So on the one hand, we're looking into how can we interpolate sequencing data, which is particularly relevant for our setup because we have initially dense sampling and later on less dense. So that would be really helpful. And at the same time, it gives you a lot of information about the dynamic modes that are present in your transcriptomic data. So it can tell you, for example, which genes are affected by circadian clock or by day-night cycle, and which ones not so much, or which ones correlate to some other process. We're also working into dimensionality reduction to see if some parts of key genes we can really model as individual variables, but whether for other groups of genes we can sort of cluster them into a group of genes that then is represented by a single variable and that we then also incorporate in these networks. And we're also looking into methods that you can actually go from the transcriptomics data and use machine learning approaches and physics-based machine learning to see what kind of equations you are still missing. So then you could use your mechanistic model as sort of priors and try to see what terms and what variables that are important you are missing. And we're exploring, like, how can we integrate these different mechanisms? For example, if you have interpolation between differently spaced data, that might actually help you to make the module finding or to make the equation learning easier. On the other hand, maybe you first need to find your modules, because then you reduce the number of dimensions for which you have to do this interpolation or this equation finding. So we are still in the process of exploring these different directions between the different approaches. But in the end, for the project, our vision is to arrive at a hybrid model where we integrate mechanistic modules for the different responses and the different organs in the plant with machine learning modules that, for example, modulate a particular connection between genes or represent a whole group of genes or modulate the environmental inputs into a gene to sort of get the best of both worlds, and on the one hand maximize understandability, and at the other hand take into account this bounty of transcriptional data that you couldn't do in a mechanistic model alone. And we wrote a review about this recently. So with that, I would like to thank all the people that are mostly on the demonstrator team that we are working with, and I'm happy to take your questions.
Okay, thank you, Kirsten. And we have our microphone throwing team operating. Got time for probably 2 questions, as long as the first one's not too long. Christine.
Thanks, really enjoyed that, and I think it's an exciting initiative. I was just wondering again, with the ODEs, if you're going to model your systems with ODEs, how do you think you'll scale that up?
Well, I think that's exactly the point that we are trying to make with the hybrid approach, that there's a limit to that, and that therefore we want to merge it with the machine learning. So I could imagine, like you have approach like a Bayesian information criterion, where you weigh whether you should make your model more complex relative to the improvement of your fit to your data, that you could develop a similar approach where you say, okay, I'm going to push a gene from my black box or from my big module into my mechanistic model if it improves my prediction by so much, but I'm also going to penalize for how big my model will be, and that in that way you try to optimize by how complex will be the mechanistic part. And yeah, to trade off between how much can you understand and predict versus how much doable is it still.